Hi, everyone. It's my great pleasure to introduce Sean Gibbons today. Sean is an associate professor at the Institute for Systems Biology in Seattle, where he moved after completing his PhD in biophysics at the University of Chicago in 2015, and then after that moved to Boston to complete postdoctoral work at MIT. Sean works on microbial ecology and evolution using both wet lab and computational approaches. And in particular, he's interested in how host-associated bacterial communities influence the health and wellness of the host organism and how these communities adapt to their individual host over the lifetime. I've known about Sean's work for a long time, and one of the things that's really excited me about his work is his effort to understand human variation in the microbiome using a really integrative approach. For example, identifying these inextricable links between human variation in the genome and the microbiome and the metabolome and how they're all talking to each other. And of interest to this audience, I wanted to point out to you Sean's 2022 paper in Advances in Nutrition, leveraging the gut microbiota to predict personalized responses to dietary prebiotic and probiotic interventions. I know that many of you will be very interested to have a look at that if you haven't seen it already. So without any further ado, it's a pleasure to offer my virtual welcome to HNRCA. Welcome, Sean. Looking forward to hearing your talk today.
Thanks, Daniel, for that really kind introduction and to everyone for this invitation. It's an honor to be with you all. I miss Boston, so I hope weather's nice over there and everyone's enjoying the transition into fall. Um, so today I'm going to talk a little bit about our work from the last few years on the microbiome and aging with a little bit of nutrition thrown in there, and maybe try to convince you all that, it's not all bad getting old. But to start off, just to give a brief introduction for those who may not be familiar with the microbiota, we are living in a microbial world, right? Us multicellular organisms have evolved in the context of having microbes in and on us. We are densely populated by microbiota. If you look at the numbers, we're about 50% microbe. There's about 40 trillion microbial cells in and on our bodies, and 30 trillion or so human cells. Depending on your last bowel movement, you may be more microbe than human. And the microbiota is a unique part of our phenotype, right? Just like our genomes, it's one of the things that makes us unique from any other person. In fact, even if you're an identical twin who shares the same genome, your microbiota are almost as distinct as two strangers. And if you look at the genetic capacity of the microbiota, they vastly outstrip us, 100 times more genes, unique genes in the microbiome than there are in our own genome. And over the course of our evolution, we have outsourced a fair amount of the functional capacity of our bodies to this ecosystem that lives in our bodies. We wouldn't be able to digest certain substrates in our diets if we didn't have our microbiota. We wouldn't. We would develop oddly if we didn't have our microbiota, as you might know from germ-free mice who are weird and have kind of weirdly shaped organs that aren't kind of what you would expect. Their immune systems are kind of out of whack. So we need our microbiomes. And it's one of the things that shapes us as we age. So I'm just going to jump straight into aging in the microbiome. This is a field that we got into probably 5 years ago at this point. Um, and when I started thinking about this question, I was reading the literature of what had been done previously. There have been several groups that have worked on this question over the years. And there were kind of two camps. There was one camp where, we were sort of managing the decline of the organism. Aging was a process in which you're kind of losing capacity, you're losing functionality with time, and the more you can forestall that loss, the better you age, right? You try to slow or stop the aging process because generally aging is bad and it's not good for us. And a lot of this work in the microbiome space was done in assisted living facilities where people had fairly poor health. They weren't community dwelling older people. And there you did kind of see that you get this kind of maintenance of a kind of younger microbiome. But then towards the very latest stages of life, you started to see the rise of pathobionts, you see the loss of potentially protective taxa, and eventually death. However, there's another set of groups that we're looking at healthier older folks, in particular centenarians, and there they saw a different pattern where these community dwelling or healthier older folks saw a lot of dynamics, a lot of change in their microbiomes, more so than people who were less healthy. You saw a loss of core taxa, the organisms that tended to be dominant in our youth, and the rise in subdominant taxa over time. So kind of maybe compensatory changes that might actually be beneficial or help us as we get older. And there was a bit of a tension between these two camps, and so our group kind of walked in to try to resolve some of this tension by looking at deeply phenotyped cohorts where we had a lot of data on the host and paired data on the microbiome. So to give a little intro to those cohorts, we looked at a cohort called the Arabelle cohort. This is from my institute. It came out of a spin-out company called Arabelle. It was a personalized medicine wellness company where individuals had their bodies quantified, soup to nuts. Their genomes were sequenced, their blood metabolomes, their proteomes, their microbiomes, including questionnaire data and clinical data. So we had all this paired information from a population of folks that spanned the entire human lifespan from 20-year-olds up through to 90-year-olds. We can split these populations into different sets based on sequencing vendors and so on to get sort of test and validation cohorts. We also pulled in a cohort of older individuals. This was the Mr. Oz cohort, and it's actually solely a male cohort. Arabelle is actually more female than male. It's 60% female. Mr. Oz is 100% male. It was originally recruited to study osteoporosis in men, and it's about 1,000 older men between about 80 and 100 years old. And there, similarly, we have deeply phenotyped data there. So, blood metabolomics, microbiome data, and clinical and health data. We also pulled in a few other external data sets like the American gut cohort to kind of have further validation for our observations. And so we sliced and diced the data a lot of different ways. And one of the strongest signatures that came out of the microbiome in our analysis was what we called uniqueness. So we sort of invented a new metric based on beta diversity. So beta diversity is sort of the similarity between two microbiomes, right? You kind of calculate a distance metric between two vectors of relative abundances. And here we're using the Bray-Curtis metric, which is just a weighted metric of dissimilarity between microbiomes. And we saw that over the decades of life, there is this sort of accelerated increase in this uniqueness score. And the way we calculated uniqueness was it's the nearest neighbor distance to another individual in the population, right? So how close are you to your closest neighbor? And the further away you are from that closest neighbor, the higher your uniqueness. The more unique you are in the population, the higher this score. And so this number is going up as we get older and older. Cross-sectionally, if you look across the population, starting in about the 40s or 50s, you see that this starts to tick up. This is true across different, test and validation sets and across very different cohorts. So here in the American gut cohort, we also see a similar increase in uniqueness. And at this point, there's been, a couple of dozen groups around the world who've seen a similar signature in the guts of older folks. One thing I'll say to caveat this a little bit too, though, is that the Arabelle cohort is a largely healthy cohort. These folks are people kind of randomly selected from the Seattle population. They tended to be a little more affluent because they were paying for this precision wellness service, and they were kind of health conscious and receiving kind of a healthy lifestyle advice, coaching intervention. If we look at the taxonomic signature of this uniqueness score, we find that it's very negatively associated with gram-negative anaerobes like Bacteroides and Prevotella, and it's positively associated with gram-positive anaerobes like the Firmicutes, and in particular, a lot of the Clostridia, sort of the butyrate producing taxa that live in the gut. So that's the dominant signature. There's actually a lot of different types of Clostridia, and so each one of us tends to kind of have a little bit of a different set of these Clostridia, whereas almost all of us have Bacteroides or Prevotella. And so this is part of what drives this uniqueness factor, is that if you have a decline in this shared core genus Bacteroides and a rise in all these other taxa that tend to be kind of more patchily distributed across the population, you then see a concomitant rise in this uniqueness score. We can look into the bloodstream and ask, what blood metabolites are associated with this uniqueness. And we find that it's a lot of protein fermentation byproducts. So fermentation of like tyrosine and tryptophan and acetylglutamine or acetyl phenylacetate, sorry, phenylacetate. So these particular molecules are higher in the bloodstreams of people with higher uniqueness scores. Um, Kind of interesting because some of these molecules are kind of toxic, right? So I think phenylacetylglutamine, p-creosyl sulfate, these have been associated with like kidney disease or cardiovascular issues. But then other molecules like indoles have been associated with lower inflammation. And in fact, I think indole has been tested in mouse studies. If you feed indole to mice, they have extended health spans. Um, so a bit of a kind of equivocal signal there in the blood. So kind of painting our microbiomes out on beta diversity space, right? If we project the similarity of different people's microbiomes onto a two-dimensional plane using principal coordinate analysis, where distance between points, points are individual people and their microbiome composition, and distance between points is sort of how dissimilar they are. We can paint this picture by certain genera. So here, Bacteroides, this dominant gram-negative genus, especially in the developed world, is more abundant out on this arm of this curve. Prevotella, which is less frequent in the developed world and is more sparsely populated here, as you'd expect, an American cohort, it's defining this arm of the curve. And this uniqueness score is enriched here at the elbow. And in addition to uniqueness, at this elbow, you also see an enrichment of these gram-positive anaerobes, butyrate producers, and organisms like Akkermansia muciniphila. So when we look at uniqueness in the aging cohort that we were looking at, the Mr. Oz cohort, we actually saw that there was a weaker association between age and this uniqueness score. So we thought maybe this is pointing towards this bifurcation or this tension we see in the literature. What if we partitioned the population by their health status? So when we did this in the Mr. Ross cohort, and at the time we had access to a few different metrics of health. One was walking speed, right? How fast can you walk? One was self-reported health, whether you say you're excellent or anything but excellent. Number of medications used, high versus low. I think the threshold here was 7. So if you had more than 7, you were high. If you had less than 7, you were low. And then life space score is kind of a metric of how much you get around in your life. We had a high and a low threshold there. And then we had a composite score where you had to be healthy according to at least 3 of the other metrics to be composite healthy. And no matter how you sliced it or diced it in the discovery and in the validation cohorts, we find that people who report being healthier show a fairly strong association between uniqueness and age, whereas those reporting having less than perfect health, those folks, we see kind of an obliteration of this signal. We don't see this association with uniqueness. So that was kind of interesting. It seems to be associated with health, especially in older folks who are healthy versus not healthy. We also had follow-up data for the Mr. Oss subjects. So there was a 4-year window between 2 visits. And so we had a baseline microbiome sample. We could partition those individuals based on whether they had high or low uniqueness, or whether they maintained high or low dominance of these gram-negative anaerobes, which is sort of anti-correlated with uniqueness. And either way, you find that people with higher levels of Bacteroides or lower uniqueness were significantly less likely to survive to follow up over this 4-year window. And if you use Cox proportional hazards regression, controlling for a number of cardiometabolic risk factors, like a lot of clinical chemistries like LDL and insulin resistance and so on. You see that, even controlling for these other factors of mortality risk, there's a significant association with high levels of gram-negative anaerobes and uniqueness. So not only is it kind of correlated with health in later life, but this seems to be predictive of survival outcomes. We're still trying to understand what is driving this signature, what is mechanistically responsible for this phenotype. We have now some metagenomic data. So the data I presented earlier was all 16S amplicon sequencing. So if we look at metagenomes, we start to see some patterns emerging. One pattern that's coming out is that molecules associated with dampening inflammation are higher, or those genes are more frequent in higher uniqueness microbiomes. And genes associated with host glycan degradation, like mucus degradation, they tend to be anti-correlated with uniqueness. And sort of makes sense, right? Inflammaging, inflammation is not great for people, especially as they're getting older. And as we get older, our mucus layer is also thinning. We're producing less mucus in our intestinal epithelium, and maybe we're eating less dietary fiber or just less in general. And these things together could lead to thinning mucus layers and also to inflammation. So perhaps this change we're seeing is compensatory to these things. Okay, so now I'm going to switch gears. That was the 2021 paper that we kind of got our feet wet in aging and the microbiome. We had another paper looking at interactions between the microbiome and drug use, in particular statins. And this becomes relevant to the prior story, which I hope hopefully you'll see here as we get through this. But basically, what we wanted to look at in this particular study, and here we're leveraging again the Arabelle cohort I was talking about before. We had seen these kinds of results from big clinical trials, right? So this is the Riddicker paper from 2016. You give about 8,000 people the same exact dose of a statin, and there is quite a large range of responses in terms of changes in LDL cholesterol. Some people, on average, about half of the people achieve the benefit that you see on the label of the drug, which is like a 50% reduction in LDL. But some people get a much larger reduction, and indeed, some people see an increase in their LDL cholesterol after, I think this is about 6 months or a year of taking the statin. And the level at which you bring down your LDL is in this cohort was associated with events. So the more you could bring down your LDL, the fewer heart attacks and strokes were being had by folks. So we wanted to maybe see if the microbiome could explain some of this variability. Another kind of poorly understood side effect of statins, and of course, the most common side effect of taking statins is kind of muscle pain. But there's another side effect, which is kind of a perturbation to glucose homeostasis. You see that people on statins have about a twofold higher risk of getting type 2 diabetes. And this is epidemiologically, even after you control for a lot of other lifestyle factors and so on. And then there's been some recent clinical trials measuring this, and they've shown that if you start giving people statins, you see that there's a perturbation to their insulin resistance. And so what's going on here? So we thought we'd kind of look at both the on-target and off-target effects of statins through the lens of the microbiota. So in the Arabelle population, we had about, what is it? Yeah, 1,600 non-users and 244 statin users. And these were validated. So we had kind of clinician information that these people were either on statins or were not on statins. For a subset of the statin users, we actually had dosing information. So for a little less than half, we could tell whether people were on a high, a low, or a moderate intensity dose of a statin. And you can see kind of the different types of statins that were being used. Atorvastatin is the most common. We had blood metabolomics, microbiome sequencing, and a bunch of questionnaire data for this population. Okay. One of the challenges for the reviewers for this paper was that we had only cross-sectional data here, Usually, if you want to look at statin efficacy, you look at the change in LDL over a period of time. But we couldn't do that. We couldn't calculate a delta, and we actually didn't know when people started taking the statin. We just had a snapshot of people who were and weren't on it. So one way we tried to get around this is we thought, okay, well, statins are inhibiting this enzyme called HMG-CoA reductase, which is the rate limiting step in the cholesterol synthesis pathway. And it stands to reason that if you inhibit this enzyme, the upstream factor is going to kind of build up. And there's an esterase that kind of cleaves off the CoA here. So we thought maybe this HMG is accumulating in the bloodstream, and it could be a marker of the sort of on-target effect of the statin that's sort of a instantaneous metric of efficacy. If we look at folks who are taking statins versus not taking statins, we see that there is this association only in the statin users, where your degree of your level of LDL cholesterol is negatively associated with the level of this HMG in the bloodstream, and there's really no association in people who are not taking statins. Um, to further kind of assess the utility of this molecule as a metric of efficacy, we looked at the dosing information, and as one might expect, as you increase the dose or the intensity of a statin, you see a progressive kind of a linear relationship between the dose and the level of HMG in the bloodstream. Um, Similarly, how low your LDL cholesterol is also associated with this intensity of dosing, significantly so, controlling for sex, age, BMI, and batch. One other way we tried to validate this was there are known genetic markers of statin efficacy. And so Tomasz Wolanski, the first author of this work, he pulled down the 9 strongest genetic um, loci that are associated with statin efficacy. And we use that in a regression model to sort of account for all the variation that could be explained by host genetics in statin responses. The top um, variant here is in the ApoE gene. And as you can see, in people who aren't taking statins, there's no difference in your HMG levels. If they start taking statins, you get a bump upward in this HMG level, no matter who you are. But if you have this T allele, which is associated with statin responsiveness, you see a bigger bump, right? So we sort of convinced reviewers that we could use HMG as this marker of statin efficacy. I should also mention that with the genetics, we were able to show, we could explain some of the variation in statin response. And we can compare that to the amount of variation that we could explain by the microbiota. And we could explain actually a larger fraction of the variation with the microbiota than we could with genetics. So to give you start giving you a picture of the microbiome and its association with statin efficacy, this is now just looking at alpha diversity or kind of species diversity in the microbiome and its association with this HMG and statin users and non-users. And what we saw is that people with higher levels of diversity showed a weaker on-target response to statins compared to similar diversity folks who aren't taking statins. Their HMG levels, or the difference in their HMG levels, are here. And the interaction effect is significant. So if you have and these are tertiles now of diversity. So this is the highest diversity tertile and the lowest diversity tertile. This is Shannon diversity, which is a mixture of the species richness and the evenness of the community. And this is observed ASVs, which is just counting up the number of taxa in the sample. So it's just species richness. Kind of counterintuitive, honestly, when we first saw it. But those people with the lowest diversity microbiomes show the biggest on-target effect of the statins, controlling for a lot of different things. And you could think, well, maybe this is because of an asymmetry of dosing between these different people. And that wasn't the case. So if we control for dose in those samples where we have dosing information, we find that there's still this negative association between diversity and HMG levels with this control for dose. So looking beyond just diversity, there were prior papers who looked at the effect of statins on people and their microbiomes. And this is from Europe, this prior work. And in Europe, it's very popular to do this enterotype clustering. And so we had to kind of compare our results to what they had found. If you're not familiar, this is just kind of a coarse graining method for clustering microbiomes into big coarse groups. Here we're using a method called Dirichlet multinomial mixture model clustering. And we get these four groups, and this is consistently what you see generally in the developed world, especially in Europe and the US, is you get four groups with these names, BAC1, RUM, and BAC2, and PREV. So kind of give you an idea of what these groups mean in terms of taxa. The BAC1 and BAC2 groups are enriched in Bacteroides, this genus that you heard about before in the aging story. And the Raminacoccaceae group is enriched in gram-positive anaerobes and is depleted in these Bacteroides. The Prevotella group is distinct in that it's highly enriched in this genus Prevotella. Prevotella and Bacteroides tend to not co-occur very much, right? Either one is really dominant or the other. And so this is a very distinct group here, the Prevotella group. And it's actually the smallest N group in our cohort, so it's actually hard to do statistics with the Prevotella group because the N is pretty small here. Between the BAC1 and BAC2 groups, one of the main things that distinguishes them is that the BAC2 group, it's been associated with diabetes, obesity. It's kind of like the bad guy enterotype. And you see that organisms like Faecalibacterium and other common butyrate producers in the human gut are really depleted in this particular group, whereas BAC1 still maintains some of these butyrate producers. So that's an overview of these enterotypes. If you look back at this PCOA I showed you before with the uniqueness elbow here, this Ruminica group is enriched at the elbow where, BAC1 and 2 are over here and Prevotella is over here. So if we look at on-target effects of statins, thinking again of HMG here versus these different enterotypes, comparing statin users and non-users are in gray. You can see that for almost every one of these enterotypes, you see a bump in your HMG if you go onto statins. Again, for Prevotella, we have a really small N, so I don't know how much I trust the stats here. But you get the biggest bump in the BAC2 group. This is sort of the bad health group, but this group is actually showing the largest jump in HMG when you take statins. This is also the lowest diversity group, so it's probably driving that diversity signal I was talking about before. And if you look at LDL levels, those statin users who are BAC2 show the biggest drops in their LDL. So you get a big on-target effect if you happen to be in this BAC2 group. Now, the downside, though, is if you look at insulin resistance, you have the biggest perturbation to your insulin resistance when you're in this BAC2 group as well. So you get a significant increase in HOMO-IR, but you see similar effects in HbA1c, in glucose, and insulin. Now, this is sort of true also in the BAC1 group. So both of these BAC groups, you see a bit of a perturbation to insulin resistance. But in the Raminacoccaceae group, this Firmicutes enriched group, there's no significant difference, right? And this isn't an N issue. Like there's actually a lot of people in this grouping. You seem to be protected from the metabolic side effects of statins if you're in this group, yet you're also seeing a significant on-target effect of the drug. We can validate this in another cohort. So that was in Arabelle. Now we look at the Metacartus cohort in Europe, and we see the exact same signature, right? You get this perturbation, you get this HMG on target effect jumps up in these groups, and you get this association with insulin resistance and Bacteroides abundance. So, how is this related to aging? Well, a lot of people as they're getting older are taking statins, right? 22% of our population were taking statins. So a large fraction of American adults are on this drug. And it seems that maintaining high dominance of these gram-negative anaerobes that was kind of anti-correlated with this uniqueness metric we were talking about before, this is associated with a greater perturbation to your insulin resistance when you take this drug, even though you're also getting a big LDL response. But having higher levels of butyrate-producing Clostridia and Akkermansia muciniphila, which I didn't have time to talk about here, were associated with protection from the metabolic effects of the statins. So this maybe gives us some hints at ways in which we can protect aging folks from things like metabolic perturbations, type 2 diabetes, Perhaps probiotics that include things like butyrate producers and Akkermansia might be useful in these folks. So overall, we wrote a perspective piece on this a couple years ago. We agree with the literature, right? There appear to be two different things going on. There are two different signatures of aging in the gut, one associated with people who are in better health, and one associated with people who are in worse health. Those who are in worse health, they actually retain a microbiome that looks more similar to what you have when you're younger, right? It's more enriched in these gram-negative anaerobes, and this perhaps isn't a good thing as we're getting older. And the folks that show the biggest deviation from the signatures we see in youth, they seem to be healthier, and they have this rise in the butyrate producers and the Clostridia. So perhaps some of these changes in the gut are compensatory. They're probably associated with healthy patterns of lifestyle, like maybe having a higher plant-based diet, a lot of fiber degraders in this healthy aging group. But we don't really understand exactly what the mechanisms are, like what are driving these patterns. And I would argue that if you want to get down into the mechanism, what's happening with the microbiome and human health, it comes down to the small molecules, right? Much of the functionality of the microbiota is driven by the production of small molecules or metabolites. Um, in a paper that Daniel referenced earlier, we actually looked at this and compared the effect of the genome and the microbiome on circulating metabolites in the bloodstream, and the microbiome beat the genome by a pretty wide margin. It was about 44% of all the circulating metabolites in the blood showed a significant association with with a genus in the microbiome, whereas less than 10% of circulating metabolites showed an association with a loci in the genome. This was, the power was limited here by about a 2,000 person cohort, but given that power, we were, there was much more stuff associated with the microbiome than with genetic variation. We don't yet have a Rosetta Stone to understand, how do you convert, microbiome composition into this metabolic output, and that's over the past few years kind of what our lab has focused a lot on, which we'll talk about here in a second. And in terms of aging, we know that there are bad guy metabolites. So this is not from our group, but this is a recent paper that just came out, I believe in Nature, where they identified a molecule called ADP heptose. And it's enriched in animals when you give them DSS, so you induce colitis. But it's also enriched in older animals. So it seems to be associated with aging and perhaps unhealthy aging, and it's higher in different diseases. And it was associated with poor outcomes in this kind of model of cancer, especially in aging animals. And this molecule is exclusively produced by gram-negative anaerobes, like the Bacteroidetes. So perhaps another good reason why you don't want to have a lot of gram-negative anaerobes in your gut as you're getting older. But there are also good guy metabolites. And I've invoked butyrate earlier in the presentation, but butyrate is it's a short chain fatty acid. It's produced by fermenting dietary fiber into these organic acids, and butyrate is probably the most potent of these organic acids. It's very healthful. It has myriad effects across the body that I won't go into all of them, but for example, it can affect host gene expression by being an HDAC inhibitor. It can affect differentiation of T cells, like it enriches for T regulatory cells and depletes Th17 cells in the intestinal epithelium. It seems to reduce systemic inflammation, help with cardiovascular disease risk, help maintain glucose homeostasis in the blood, and so on and so forth. But the problem is, if I fed 10 people a banana, very different amounts of butyrate come out the other side. Generally speaking, also in the developed world, we are very underexposed to dietary fiber. Historically, we probably ate 100 grams of dietary fiber a day, and now we're lucky to get 10 grams a day in the standard American diet. And so how, for this limited fiber budget that we get as humans, how can we predict optimal levels of these good guy metabolites to maybe improve outcomes, help with aging and inflammation? So we developed a tool called MyCom, which we kind of see as our Rosetta Stone for converting microbiome composition into these metabolic outputs. I won't describe the method in too much detail, other than you can think of it as a digital twin, if you will, of your gut microbiome metabolism. You can use sequencing data from an individual to constrain who is in their model, like what organisms are there and in what relative abundances. We map those taxa to a database of curated genome scale models called Agora, and we build sort of a representation of their microbiota. We also have to build in a representation of their diet. So we constrain the model with a diet. We have a few pre-computed ones, but you can also create custom diets. That diet is run through the human metabolic model, Recon 3D, where we strip the diet of all the components that are absorbed in the small intestine and consumed by us before we feed them to the colonic milieu. And we inject a few things we know the host are putting into the diet, like mucus and bile acids. Basically, you run these models, you get predictions of the growth rates of all the taxa in the system, and you get predictions of cross-feeding interactions, the net consumption of metabolites from the diet, the net production of metabolites that are available to the host. And we call these things microbial community scale metabolic models, or MCMMs. And this whole modeling software platform was developed by Christian Diener, who was a research scientist in my lab and now runs his own group at the Medical University of Graz in Austria. And the first kind of practical use case we put to this modeling platform was to predict short chain fatty acid production, like butyrate, right? If we feed a bunch of people a banana, can we rationally predict what comes out? Maybe some people should eat a banana and some other people should eat a tangerine. Can we predict this? One of the challenges to short chain fatty acid production is that it's really hard to measure. If in the gut, butyrate is the primary carbon and energy source for enterocytes. So as soon as it is produced, it is consumed by the intestinal epithelium and converted into biomass and carbon dioxide. And so that you get of these things in the bloodstream or in stool are noisy proxies for the in situ production rate. So the way we got around this is we had poop soups. So we had about 40 people. We recruited from the Fred Hutch. We got really, really fresh stool. The student would run down as soon as we had a poop drop. They would get the poop back within 15 minutes to our anaerobic chamber. We would homogenize that stool in a buffer, kind of liquefy it. And then we could add dietary interventions like a dietary fiber, inulin or pectin, and then do time series metabolomics to measure the production flux of these various molecules. And so this paper was from this last year, where we showed that we could get some quantitative traction in predicting butyrate and propionate, who are some of the most potent of these short chain fatty acids. We get an R value of about 0.6, which by no means is a perfect prediction here, but it's pretty good, especially considering that these are mechanistic models. There was no machine learning going on here. We're not fitting anything. So this is just purely a mechanistic prediction, and we're getting reasonable accuracy. Just to compare this to other machine learning models that are out there for kind of translating microbiome composition into host phenotypes, this is from a few years back. You're probably familiar with this Zivi et al. Paper from the Siegel lab in Israel, but they had an Israeli cohort. They measured their microbiomes, clinical chemistries, and a few other things, along with diet tracking over the course of time, and were able to build, I think it was a gradient boosted decision tree model to predict post-meal glucose curves. This is the area under that curve predicted versus measured, and they get a R value of about 0.68, which is a little better than how we're doing. But again, this was trained, and we expect that machine learning models will perform a little bit worse when you apply them out of the training set, whereas mechanistic models, they should perform about the same no matter where you apply them. So that was in vitro validation. We can also look at a sort of in vivo validation, right? Like I said, we couldn't really measure butyrate in vivo, but you can measure the downstream effects of what it would look like in the bloodstream if you were to produce more butyrate over the long term. So in this Aravel population, where we had paired clinical chemistries and stool microbiome data, we could build their models. We fed everybody in the population a standard Western diet in silico, which is not correct, right? We know everybody has slightly different diets, but it was easy to do because we didn't have diet information. And we regressed those predictions, say butyrate and propionate production, against about 130 of these clinical chemistries, and about 30 or 20 or 30 of them came out as significant. The stars are denoting statistical significance. We had levels of butyrate measured in the bloodstream, and none of that was associated with none of these clinical chemistries. So kind of the blood measures of butyrate was sort of like white noise. We saw no associations with predicted propionate for any of these chemistries. To draw your eye, the blue arrows here are showing lipid markers of health. So if you were predicted to produce more butyrate on a standard Western diet, you tended to have lower LDL cholesterol, lower triglycerides, etc. You were also predicted to have better insulin sensitivity, right? So if you produced more butyrate, you had higher insulin sensitivity or lower insulin resistance. And then markers of inflammation like CRP, if you produced more butyrate or predicted to produce more butyrate, you had lower levels of blood CRP. So this sort of makes sense. These are the kinds of effects we would have expected. And so our predictions seem to have some traction in predicting host phenotypes. So assuming this kind of works, you can start playing some games, right? Like we can run a clinical trial in silico. We can take this Arabelle population and we can flip them between a standard European diet, and this European diet was based on, I believe, the Austrian diet. So it's kind of more similar to the American diet, and a vegan high fiber diet. When we do this, we can see that we can indeed turn up butyrate production across the population. So this is predicted butyrate, and we see we can push it upwards with a high fiber diet, but the peak is very broad. And there are a fair number of people who are non-responders to this dietary intervention. They don't see a big jump in their butyrate production when they switch. We also see a few regressors who actually produce slightly less butyrate when they switch to a high fiber diet. So we wanted to see in our model, so this we haven't done this in a trial, right? This is not experimental data. But in the model, if we could add two different prebiotics, either inulin or pectin, or a probiotic organism, so this is Faecalibacterium presnitzii, which is a prevalent butyrate producer, can we rescue these non-responders with a simple co-intervention on the backgrounds of these diets? So now this is the result of that. The heat map, the heat is showing butyrate production. Every column is an individual person. The top here is coloring by whether they were a regressor or a non-responder, and the black boxes are outlining the optimal intervention for that person. And so if you scan across, you can see that there was no single intervention that rescued everybody. Different people seem to be rescued by different interventions. For the regressors, frequently just taking a prebiotic fiber on the background of a diet was enough to really push up your butyrate production. But for many of the non-responders, it didn't really matter how much fiber you fed down their system. If they didn't have the genetic capacity to produce a lot of butyrate, they just didn't make much. So it required the addition of a probiotic organism like Faecalibacterium in order to elicit butyrate production from their system. And so this is something we'd love to test prospectively in a trial, but we think we have, kind of a digital twin platform here for predicting personalized responses to diet. So getting back now to aging, as I wind up here, I'll kind of update on some of that aging work and where we've gone with it next. If we look at, papers that have come out more recently in this space, we find that they're pretty convergent with what we found. So in this paper, this is a Chinese cohort, and they're looking at frailty in older adults. They found that frailty was generally negatively associated with Clostridia, these butyrate producers, where it was positively associated with gram-negative anaerobes and with bacilli, which is another class of firmicutes that tends to be a signature of, not great health. And if we kind of apply our metabolic modeling, so this is just a hairball picture of one of these MyCom models. So this is one person's microbiome. You can see all the different metabolic exchanges happening. The regions of high density are individual taxa, and you can see kind of internal fluxes and cross-feeding interactions between taxa. If we apply these models to our aging cohort and look at uniqueness, we see that there is this significant association between the predicted butyrate flux, how much butyrate they produce, and their uniqueness score. All right, and this could potentially be one of the drivers of why are people with higher uniqueness seeing better outcomes or better health late in life? Maybe they're producing more butyrate. In the Arabelle cohort, we actually were able to calculate frailty metrics. So this is now work in collaboration with Dr. Alice Kane, who's an aging researcher who we hired a couple years back here at ISB. She's an assistant professor, along with Dr. Noah Rappaport from the Hood Lab and my postdoc, Alex Carr. And so here, Alice calculated frailty metrics based on lab values. So she has a weighted scheme for making this metric from labs, clinical chemistries, and also from self-reported questionnaire data. And then the merged frailty index is a merged score that takes both self-reported questionnaire data and lab values. I can't tell you exactly how they were calculated, but Alice can, and we'll have a preprint soon that can kind of explain all of this. When we look at the taxa and Arabelle that are associated with these frailty scores, again, this makes sense. Here we're controlling for age, sex, body mass index, and the first four principal components of genetic ancestry. We see that a lot of butyrate producers, gram-positive organisms are negatively associated with frailty, and you see kind of bacilli and gram-negatives are positively associated with frailty. We can look back at these enterotypes that I talked about in the statin study. And so if we look at enterotypes, we find that this merged frailty index, on average, it's higher in the BAC2 group. So people who have this kind of bad enterotype, they tend to have higher frailty, which is kind of what we expected. And this Rominococcaceae group that's enriched in the butyrate producers, it has the lowest level of frailty. We can also look at this UniFrac. This is, I believe, the uniqueness score based on the UniFrac metric. Uniqueness is highest in this Rominococcaceae group. And butyrate production predictions from our metabolic models are also highest, as one might expect, in this Rominococcaceae group, and lowest in this BAC2 group. And looking at things associated with butyrate production in the microbiota, again, this makes sense. If you're a butyrate producer, you're positively associated with butyrate production, and you see that kind of gram negatives and bacilli are negatively associated. Okay, so this final piece for this Arabelle cohort that I'll talk about is a longitudinal analysis. So one thing I didn't talk about much in Arabelle is that individuals who were recruited into this cohort were given personalized coaching on healthy lifestyle. So they were told to eat better and exercise more, kind of on average. It's a little wonky because we don't know exactly what every person did, but we know that on average they were being called once a month or once every couple of weeks, I think, by a nurse or a nutritionist to counsel them on their behavior. And if you look at their baseline versus like a 6-month follow-up, we can calculate the change in their frailty index, right? So how much did their frailty index change over that 6-month to 12-month window? On average, pretty much everybody showed a reduction in their frailty. So people are getting healthier with this intervention, is kind of what we would expect. But interestingly, and this is sort of a sex-specific effect, in the males, we see in the BAC2 group, they get the biggest bang for their buck here. So the males who go on this healthy lifestyle intervention, they can bring down their frailty the most. This kind of makes sense to us because the BAC2 group, those individuals probably start off with the worst diet and lifestyle, and so they probably have the most to gain from these types of lifestyle changes. Whereas other people, might need to do other things like take a statin or do something a little more pharmaceutical in order to get changes to happen. So maybe you could partition people by their microbiome status to kind of target lifestyle versus other types of interventions. The very last thing I'll quickly talk about is a mouse experiment that we're helping out on. This is Alice Kane's work, actually, from her postdoc. She did a supplement trial in mice where they gave them NMN, which is one of these NAD precursors, long-term supplementation of NMN over the course of the lifespan of these mice. So, if you look at the beta diversity of the microbiome, and this is work driven by Christian Diener and Alex Carr, there's not a huge difference in males and females between treatment and control, but it's significant. There's a bit of a shift in their microbiota in kind of overall beta diversity space. But there was one screaming white hot signal that jumped out in the genera or in the actual species that were present in the system. This one organism, a Narrowtruncus collihominus, which is a gram-positive butyrate-producing organism, it was 10 times enriched in the treatment group for both males and females. And it was just a very, very clear signal that persisted throughout the time points. And this organism was significantly associated with the frailty index that was calculated for these mice. In the females, it was non-significant in the males, but the sort of trend was very similar. And this is controlling for age. So, not only is it kind of increased with this NMN supplementation, it is itself associated with frailty. Um, and if we connect this back to our human data, we see kind of convergent signatures here. So Anerotruncus collihominus, again, it's this gram-positive butyrate producer. If at some baseline time point, the abundance of this organism is higher, the time to death for these mice was longer. So, it's associated with longevity in the mice. And if we look at, the most anti-correlated taxon in the data set with time to death, it was this Bacteroides uh, sechimuris, maybe is how you pronounce that. Again, a gram-negative anaerobe, and it's negatively associated with longevity. And this fits very well with our initial study where we kind of see this pattern where maintaining high levels of gram-negative anaerobes, maybe not such a great thing. Having more of these butyrate producers, probably a good thing. Okay, so take-home points. Yeah, maintaining high dominance of gram negatives into old age might not be great. We don't exactly know why yet. There could be a host of reasons, but this seems to be a clear signature out there in the data. Having higher levels, on the other hand, of butyrate producers and Akkermansia are associated with health in older, especially industrialized populations. We don't know as much about non-industrialized populations, perhaps due to beneficial effects on insulin sensitivity and reduction in inflammation. We think that our MCMM-based optimization of prebiotics or diet could help maximize things like butyrate throughout the lifespan, and this might be especially important in older individuals that are trying to manage their levels of inflammation and reduce inflammaging. But the last point I'll end off on here is that I don't think it's a good idea to take fecal transplants from 20-year-olds and put them into 80-year-olds, right? This is going back to the title of the whole talk, which is should we rejuvenate the gut microbiome? I would say the evidence points towards no, right? The 20-year-old microbiome, highly enriched in these gram-negative anaerobes, throwing that into an 80-year-old, that might actually do more harm than good. And I think targeted approaches are probably the way to go here. So with that, thanks to all the micronauts in the lab who made all this happen, our collaborators, thanks to the funding sources, and thanks to all of you for listening.
Thank you so much, Sean. That was terrific. I want to remind everybody to please put your questions in the Q&A, and I will read them out. I will just, the questions are already appearing. I think there will be lots here. Let me just start with a kind of big picture question. And that is if you're beginning to get a sense of how much of the variation that you see and the sort of impactful variation is determined early in life from the mother at birth or from genetics, such that changing your diet in middle age isn't going to make a difference, and you can eat junk food for 50 years and can you then switch, or are we all sort of set from our very young age? When is the best time to be really focusing on a healthful diet? So it's sort of big picture question.
That's a great question. And I kind of don't exactly know the answer, but it does seem to be true now that we've looked at a bunch of intervention studies with diet that the microbiome is frustratingly difficult to change, right? Its composition is very stable in adults, and even kind of longer term dietary shifts don't have as much of an effect as you might expect on the overall composition. So we do seem to kind of lock in to a microbiome composition in adulthood. It does appear that it is modifiable over the very long term with persistent lifestyle and diet changes, as some work in like immigrant populations has shown. But it's tough. It's tough to change it. But one of the things that we think about now with the metabolic modeling is, you come to me with the microbiome that you have today, and what can I put into it to optimize the outputs, right? So the precision nutrition and precision diet, I think, become very important there. And we find that even people with kind of lower diversity or what you might think of as dysbiotic microbiota, they often respond really well to diet and lifestyle changes, like you saw in that back two group, even though you're not seeing a huge shift in the composition of the microbiome. So I think, what there are ways to kind of modulate health just by kind of taking the microbiome you've got and putting the right things into it. And finally, you can really upturn the composition of the microbiome and shift it around by whacking it really, really hard. So you can give chemotherapy or antibiotics, and do this over and over and over again, do fecal transplants. This can kind of whack it around a bit, but it's a very coarse instrument. We find that there are sometimes holes in people's microbiomes that can be filled with specific taxa, but in order to know what probiotics might be optimal for an individual person, you kind of need to build a simulation of their metabolic niche space to kind of know where to plug that hole. And so we've been thinking about and working on precision probiotics using our metabolic modeling tools.
So maybe we shouldn't believe everything that we hear about on TikTok from the simple answers that are going to make us healthy by changing our gut. I'm going to read two questions to you because they're closely related, and hopefully things won't get too confusing. So the first is from Shangdong Wang, who says, "Excellent work and presentation. Thank you. I was wondering if maintaining a lower Bacteroides dominance into older age or lower levels of its distinct plasma microbial metabolites, or both combined, could predict survival outcome between male and female in centenarian populations." And then the second question from Alice Lichtenstein, in terms of statins and insulin resistance, it appears you always adjust it for sex. Have you looked at your data to see if there's a sex-specific difference in response? Now, I have the same question regarding the butyrate producers in humans. Are there sex-specific differences?
Hmm. Good questions. Gosh, let's see. So sex-specific differences. If we look, I think one of the more edifying analyses we've done recently has been Analysis mouse study. And in that analysis, there are these very large sex differences. So we see that, changes in the microbiome appear to be more associated with outcomes more in the males than in the females. So for whatever reason, yeah, the males are getting more of an effect from Coleohominus or the molecules it's producing. So that's interesting. I don't know exactly what's going on, but we're trying to dig into it. In humans, it's an interesting question with the statins. We didn't segregate the analysis and just analyze the males and the females, partially because for the males, we kind of had low N for some of these groups for the statin users. So we were a little power limited there. Um, but that's something we should look at because I think there's probably something going on there between the sexes. In butyrate production, in human beings, we don't see a ton of difference in the microbiome based on sex. You can kind of detect it if you have like thousands of people. You can see a very weak sex-specific effect, controlling for other covariates. But generally speaking, male and female microbiomes look pretty similar in humans, as opposed to mice, where you see a larger differentiation between male and female mice by sex. But it does seem that the metabolic effects and responses to things like butyrate that are being produced by the microbiome, those are probably more likely to have a sex-specific effect. how that's driven, I'm not sure, like hormones or what have you, different receptors on intestinal cells. But I would suspect that there are the responses are sex specific, whereas the composition is not so much.
And so, and briefly, just to come back to the first question, is it possible that the microbiome could account for some of the differences in in the sex bias in centenarians, for example? We know that way more women are going to make it to 100 than men.
Again, that's a great question. I don't know, but I suspect probably.
Um, the next question I wanted to ask, I know we're short on time here. Russell Teichman says, fantastic talk. There are a lot of niches of organisms which might be influencing blood metabolites. Any speculation on the role of non-gut or non-lower gut organisms in your prediction models, thinking specifically of oral organisms that could impact blood levels?
Well, that's a great question. I usually think of it as like a mass effect, like total biomass. If you look at the amount of biomass of bacteria that are in the human body, it's like 99% or more is in the gut, in the lower colon. And though that 1% is spread across the rest of the body, the small intestine and the mouth probably have the next, and the skin, I guess, probably have the next largest amounts of biomass. So I do think that there are metabolites produced by oral microbiota that are affecting the host. But if you think about the total mass amount of metabolites being produced in the human body that are of microbial origin, 99% of it is happening in the colon.
I'm going to ask one more question. I know we're at time, and hopefully you can stay on.
Yeah, I don't have a hard stop, so that's
Okay,
okay.
great. Um, so from Chao Chang Lai for the statin study, he and others have shown that statin medication contributes to type 2 diabetes risk via DNA methylation at ABCG1. Have you done any epigenetic analysis with gut microbiomes? Any work on epigenetic changes related to type 2 diabetes and aging?
We have not done that. I mean, although it is known in the field that the microbiome affects epigenetic decoration of the genome. So, like I mentioned before, butyrate is itself an HDAC inhibitor. So we know that the microbes can affect host gene expression and epigenetics. Um, the other piece I'll add to this, because I don't know exactly how to answer that question, is that there was a group in China who did a follow-up study after our study, because ours was just cross-sectional. We didn't actually do an intervention. So this Chinese group ran a statin trial, taking new statin users, giving them the drug for the first time, and then they observed that those individuals showed, higher insulin resistance after a few months of taking statins. They observed similar patterns to what we were seeing as far as like microbiome and responsiveness. We also noticed in our blood metabolomics there was a metabolite called ursodeoxycholic acid that was seemed to be protective of this insulin resistance phenotype. They actually used a drug called ursodiol, which is a clinical intervention for, I think, bile secretion, but it also can lower your LDL cholesterol because it increases the biliary secretion rate and you're losing more cholesterol through pooping out bile acids. And so if they gave a subset of their cohort ursodiol with the statin, they were able to maintain the LDL lowering effect of the statin, but the metabolic effect was completely abolished. So those patients showed no insulin resistance when they were taking ursodiol. So that was kind of a cool addition to the story.
So I'll try and squeeze in a couple more questions from Lucas Hopper. Do you think healthcare providers appropriately monitor and encourage healthy microbiomes? Do you see a path to clinical application here where there's actionable data that a clinician could collect from a patient about the microbiome that would help in healthcare?
It's a great question. I mean, that's what I hope the future is, but I know it's expensive and time consuming to collect stool samples and sequence those samples. Maybe in the far future, in the next 10 or 20 years, it'll become so cheap that it's just trivial to be able to do it. But I think currently it's not something that the healthcare system could normally do. But that being said, it depends on the clinician. But it's, I think, pretty well known that, taking things like Metamucil are good for you as you're getting older, that having a kind of whole food, more plant-based diet is probably better for you, especially as you're aging. And so this is kind of broad advice that you can give everyone without needing to know anything about their microbiomes. Eat more fiber and drink some fermented foods, exercise, those are kind of the main ways to stay healthy. But I think for more precision stuff in the future, I'm hoping we'll get there, but I think costs need to come down. Oh, you're muted.
One last question, and apologies to those whose questions I haven't gotten to. This is from Jean Flitty. I have a question regarding the relative stability of the gut microbiome in adulthood in situations of repeated antibiotic use. Does the microbiome tend to go back to its original composition, or is it being remodeled with antibiotic courses?
That's a great question. I have a student actually working on this right now in the lab. Generally speaking, you see a pretty rapid recovery after you stop taking antibiotics. Within a month or so, you're bounced back to pretty close what the alpha diversity and beta diversity was of your original microbiome. So you look like you did, but there's going to be some differences. And what those differences tend to be are you're going to lose a few bugs that were really low abundance at the onset. So oftentimes people will lose things like bifidobacterium or lactobacillus, things that might be kind of in the upper GI, like small intestine, upper colon. Their populations can bottleneck and go to extinction. And when you recover, they're gone. They've gone away. They would require, recolonization, maybe probiotics in order to recover those populations. But the most of the abundant taxa bounce back. And there does appear to be kind of a longer term successional process that's occurring that takes months to years to recover from, where you initially see kind of a higher dominance of the gram-negative anaerobes that we were talking about, and the sort of late successional phase of having higher levels of the gram-positive anaerobes, the Clostridia, that takes a little bit longer for recovery from antibiotics. And then if you kind of do this recurring treatment of antibiotics, that just kind of keeps whacking the system. You keep getting extinction events happening, and you keep forestalling that late successional recovery. And we think that that has negative effects on metabolism.
Okay, one quick last question. I see that almost everybody is still on. You've clearly captured everybody's attention here. Just a quick question from Kritika Vashista. So you talked about aqueous metabolites, and she's interested in whether you've looked at the relationship between lipidomics and the gut microbiota.
So in that 2022 paper in Nature Metabolism, where we look at kind of interplay between the genome, the microbiome, and the blood metabolome, we used the Metabolon global panel, which includes a lot of lipids, actually mostly lipids. And there's a bunch of associations. So you can actually look at the supplement from that paper, and we have all the regression coefficients and all the significant hits between what was associated with the genome or the microbiome. One interesting thing that came up were a lot of sphingolipids and ceramides were very strongly associated with variation in the microbiome. Um, and we haven't had a chance to dig too deeply into that. Another thing to mention too is there were some interaction effects with the genome. So we could see that the relationship between some of these lipids and microbes would be different on different genetic backgrounds, depending on what variant you had.
Terrific. Thank you again, Sean, for a fascinating and very thought-provoking talk. This is really an exciting time. I think your work is really helping to lay the foundation for what I too hope will be a very different way of practicing preventative healthcare in the future. And thanks to everybody for sticking around. Really enjoyed your seminar and hope
Thank
to
you.
run into you soon.
Thanks for the great questions. It was a pleasure. Good to see you, Daniel.